All right, today we are tackling one of the absolute cornerstones of biology, the Central Dogma. This is the story of how the code that's written in your DNA literally becomes, well, you. So let's get right into it. Really think about that for a second. Every single cell in your body, from the ones in your brain to the ones in your big toe, they all have the exact same master blueprint, your DNA. So how on earth does that one set of instructions manage to create so many different parts and keep the whole system running? That's the big mystery we're going to solve today.
And here's the answer, this really elegant process. You see, the central dogma is all about how information flows in one direction, from DNA to a middleman called RNA, and finally to a protein. Think of it like a massive factory. The master blueprint, that's your DNA, is kept super safe in the head office, the nucleus. But to actually build anything, you've got to get those instructions out to the factory floor. This is the process that makes all of that happen.
So, let's start at the very beginning, with the master blueprint itself. This is the source code for everything, and it's kept under lock and key in the cell's head office.
This incredible molecule, DNA, holds every last instruction needed to build and run you. But here's the catch. It's way too valuable to risk getting damaged, so it never, ever leaves the safety of the nucleus. And that creates a pretty big problem, right? How do you get the building plans to the construction site out in the cytoplasm if the blueprint can't leave the office?
Well, the solution is just brilliant in its simplicity. You make a temporary, disposable copy. This whole process of creating a portable message from the DNA blueprint is called transcription.
And this working copy, the RNA, is a little different from the DNA master. For starters, it's single-stranded, which makes it a lot more flexible. It also uses a slightly different sugar in its backbone. And here's a key one. Where DNA uses the base thymine, or T, RNA swaps in a different base, called uracil, or U. You can think of it like a different dialect. The words are just a little bit different, but the message itself gets across perfectly.
The cell is incredibly efficient. It doesn't just copy the entire DNA blueprint every time. That'd be crazy. Instead, it only copies the specific gene, or the specific recipe that it needs at that moment. And it knows exactly where to start thanks to a special sequence on the DNA called the promoter. It's basically a little signpost that says, hey, the recipe for this protein starts right here.
The real hero of this step is an enzyme called RNA polymerase. This thing is a molecular marvel. It latches onto the DNA at that promoter, it unzips the double helix just a little bit, and then it zooms along one of the strands, reading the sequence and building a matching or complementary strand of RNA. It's like a high-speed, high-precision photocopier for your genes.
So the whole process just chugs along from that start signal to promoter all the way until it hits a stop signal, called the terminator. And what you're left with is a complete single-stranded copy called messenger RNA, or mRNA for short. This message is now ready to safely leave the nucleus and make its way out to the factory floor.
Okay, so our mRNA message has arrived on the factory floor, the cytoplasm. Now it is time for the second major step, actually reading those instructions and building the final product. And this process is called translation.
But here we run into a kind of language barrier. The message written in mRNA uses an alphabet of just 4 letters. The base is A, U, C, and G. But the final product, a protein, is built from a totally different alphabet, one with 20 different letters called amino acids. So how does the cell manage to translate from a 4-letter language to a 20-letter language?
The solution is to read the message in 3-letter words. Each group of 3 bases on the mRNA is called a codon, and each codon points to one specific amino acid. This is so important. The cell doesn't read letter by letter, it reads word by word, in triplets.
And this chart right here is the key. It's the genetic code, basically a universal decoder ring for all of life. So for example, if you look for the codon AUG, you'll see it codes for the amino acid methionine. But it also acts as the start signal to begin building. And then codons like UAA are stop signals. The most incredible part? This exact same code is used by practically every living thing on our planet, from a tiny bacterium to a giant blue whale. It's amazing.
So, to make this all happen, we need a few key players on our cellular assembly line. First, we've got the mRNA. That's our instruction manual. Then we have the ribosome. That's the actual factory machinery that reads the instructions. We have transfer RNA, or tRNA, which acts like a fleet of little delivery trucks bringing in the right parts. And finally, those parts are the amino acids, the building blocks we're using to make our protein.
The whole construction process happens in 3 main stages. First, the machinery gets assembled in a step we call initiation. Second, the protein chain is actually built during elongation. And finally, the finished product gets released in a step called termination.
Let's watch it happen. In initiation, the 2 pieces of the ribosome factory come together and clamp down onto the mRNA message. They then scan along the message until they find that AUG start codon. That's the official signal to get this assembly line started.
Now we get to the heart of it all, elongation. The ribosome starts moving down the mRNA 1 codon at a time, almost like a reader moving across a ticker tape. And for each 3-letter word it reads, the correct tRNA delivery truck shows up, carrying the one specific amino acid that matches that codon. It drops it off, and the growing protein chain gets longer and longer, 1 amino acid at a time.
Finally, the ribosome gets to a stop codon on the mRNA. This is the signal that the job is done. A special protein called a release factor comes in and binds, and that causes the whole complex to just fall apart. The ribosome lets go of the mRNA, and our brand new, freshly built protein floats away, ready to fold up and get to work inside the cell.
Now, this whole system is just breathtakingly accurate, but it's not absolutely perfect. So what happens if there's a typo, what biologists call a mutation, in that original DNA blueprint?
This is such a powerful example. On the left, you see the normal code for hemoglobin, the protein in our red blood cells. But now look at the right. Notice that one single letter is changed in the DNA. A T has been swapped for an A. That one little typo changes the mRNA codon, which then causes the wrong amino acid to be brought in. This single, tiny change in the blueprint is the entire cause of a devastating condition known as sickle cell disease.
So the big takeaway here is that the accuracy of this whole information transfer process is absolutely vital. Just one typo in a genetic library that's billions of letters long can have these profound, life-altering consequences.
And there you have it, the complete journey from the master blueprint, DNA, locked away in the nucleus, to a temporary RNA copy made through transcription, all the way to translating that copy into a functional protein, the real workhorse of the cell. That flow, in a nutshell, is the central dogma.
This fundamental flow of information is what underpins all of life as we know it. But it does lead you to one last, really fascinating thought. Now that we're developing technologies like CRISPR that can let us go in and actually edit the master blueprint itself, what could that mean for the future?